Hi, my name is Markus List and welcome to the tutorial of HiTSeekR. HiTSeekR has been developed for the analysis of high-throughput screening data. High-throughput screening is characterized by sample size, so we have large to ultra-large throughput where many experiments are performed in parallel, which has been made possible by the miniaturization of cell culture assays and by the introduction of robotics that allow you to automate a lot of the liquid handling. Now, challenges in the analysis of high-throughput screening data lie in signal variation. So we have many different types of biases. We have batch effects, plate effects, positional effects, and time effects to consider. And therefore, we need to make sure that we choose for each data set the most appropriate normalization method. This, however, has been challenging because there was no really user-friendly tool that could do that. Therefore, we were motivated to develop HiTSeekR. And HiTSeekR supports three different types of high-throughput screens. It supports gene silencing, which is typically done with RNA interference, and more recently with CRISPR and Cas9, or microRNA inhibitor or mimic screens, as well as the more traditional small compound and drug screens. The goal in each case is the identification of active samples that are also called hits. Moreover, what is novel in HiTSeekR is that we don't only identify the hits, but we also connect this to systems biology. So we try to figure out which are the deregulated pathways that can explain the phenotype that we observe. For this tutorial, we'll be looking at example data that has been published in Nature Scientific Data by Katrina Falkenberg and colleagues in 2014. Here we have a study on genome-wide functional genomic and transcriptomic analysis for genes regulating sensitivity to vorinostat. Here's a quick overview of that study, where we can see that they have as a baseline an siRNA screen and a microRNA mimic screen. For this tutorial, we'll be focusing on the microRNA mimic screen, but both screens have been included as demo data sets, so feel free to play around with that. So is this study interesting? Vorinostat is a relatively new therapeutic agent that has been used in cancer treatment. However, it turned out that while this is quite a promising new class of drugs, there are many patients where this actually doesn't really work very well, and the question is why. So a little bit more about the study design here. For both types of screens, we actually have three different types of readouts. And the authors separate that into the plus drug arm, where they added vorinostat. And here we have a cell viability readout with cell titer fluor and an apoptosis readout with Kaspar's glow. So why do we need two types of readouts here? The important thing is that we want to observe everything that kills the cells quickly, and that is covered by the cell viability. But we also want to find effects that kill the cells slowly, and this is generally apoptosis. Now we also need a minus drug arm to be able to find those genes and microRNAs that kill the cells even without the drug. And therefore we use a classical DAPI staining here. So now let's move over to HiTSeekR, where we can look at this. In the first page, you are offered the choice of screen type that you want to analyze, or if you're here for the first time, you can also click here on the tutorial button and you will get to the HiTSeekR project page that has an introduction and pages for each type of the three different screen types. But let's go back and select the microRNA screen type that we want to analyze, and then we get to the input page of HiTSeekR. Here again you have a choice to make, and your choices are to either select a demo dataset here on the left side, to upload a new dataset here in the middle, or to download a dataset from PubChem via its assay ID. So for this tutorial we want to select a demo dataset, and what we get now is a preview of the data where we can see if it has been parsed or loaded successfully, and we can see which columns are already included. So if you upload your own dataset, it's very important that you click on Show File Input Options, because this tells you how the columns are mapped to the properties that HiTSeekR understands. For the demo data set, this is not so important because the column mapping is more or less done automatically. But if you look very closely, you can also see if there are problems. For example, here, okay, we're missing one of the three readouts, which is the DAPI cell count. Okay, now that we have included that and all the other properties look fine, we can also decide whether we want to log2 transform our data, and by default this is true and makes also sense. And we want to include B-scores. So this takes a little longer to compute, which is a problem if you have really large screens. But if you suspect that there might be a positional effect, then this is important. Now we click on process raw data, and you see that actually something's happening. And we see three new tabs popping up here. One being quality control, normalization effect, and hit discovery. So if you already know your screen quite well, you can jump directly to hit discovery, but typically we recommend to move over to quality control. That's also why we have this button here. In quality control, you have a number of diagnostic plots that tell you something about the properties of your screen. Here we have only two plots, and that's because in this screen we have neither replicates nor positive or negative controls. If we had those, we could actually look into more properties and also apply more methods. But here, all we have is the plate signal variation, that for each plate in a box plot tells you how big the variation is and if there are big differences between the plates, because if you have a large number of plates, then you expect the means of those plates to be roughly aligned. And this seems to be the case here. Then we have row and column effects. On this page, we're looking at positional effects. So the problem is, if you have more evaporation on the outer wells in a plate, this introduces a signal bias. And here we have the mean signal over all plates for each row and column. And if this evaporation would be a systematic problem, you would see that here in a U shape. However, we don't see anything like that here, so we assume everything's fine. Then we move over to studying normalization effects. Here you start out with a table, and this table is basically the process table that has all the properties that you selected before, plus all the scores that have been computed by HiTSeekR. You can then move on to plate signal variation, which is the same plot as before, but now you can try out different normalization techniques and see how they affect the distribution of the data. But since we only have 3 plates here and they seem already quite well aligned, there's no big effect. So let's move over to whole screen scatter plot. This plot allows you to have a closer look at the actual hits. You will always see here a black bar is the mean value, and you see most of the values basically surround this bar. These are the values where you don't expect an effect. Then you have what you might call outliers down here, and these are actually the samples that show an effect. And these are the ones that we are interested in. And you, as a user, typically want to achieve that these are separated very well from the bulk of the data. Now what you can already see here is that the first row, which corresponds to the cell titer fluoro assay, has very distinctive hits that you can easily spot. Now, if you look at the Caspars activity readout, you see there's a lot of variation, but it's not really clear if these are actually hits or not. And then here we have very little variation for the DAPI staining, but also only very few samples that show a clear effect. Okay, so let's move on to signal distribution. Here we see a density plot for each of the readouts, and you see that they are actually quite different. And this is also something that you could already guess from the whole screen scatter plot. Basically, the only screen that gives you a reasonable number of hits here is the cell titer fluoro screen. Now, we can also check out what happens if we normalize these data, and you see if you apply a set score, these 3 types of readouts already become a lot more comparable, so we can actually analyze them together. Now let's try the robust set score, which is based on the median. It still looks a little bit better. We see there are not really any hits here, very few here, and some more here. Let's also check out the B-score. Yeah, basically looks the same because we don't have a positional effect. So robust set score it is. And we can also check out the QQ plot, which tells you if the data is normally distributed. In that case, you would expect a line here, and looks quite fine. And for cell titer fluorescence, we have a large number of samples that show an effect. We have this curve here, right? But if you compare it to the raw data, we can see here, in particular for the Caspar screen, where we have this S shape, that the normalization actually helps to make this data better, more resemble a normal distribution, which is better for the analysis. So let's continue then with hit discovery. In the background, now HiTSeekR is mapping the microRNAs to microRNA families. That always takes a little while. And after this is done, you might feel a little bit overwhelmed by the number of options here. So let's go through it quickly, starting maybe with the bottom, where you see the summary of the hits that have been found with the current settings. And we have here a category column that tells you whether the found hit is a suppressor with the red S or promoter with the blue P. Then there's also a plot, a scatter plot that shows you the same hits but ordered, so you can see them very easily. And we have a heat map of the entire screen, and this is nice because it allows you to see if some hits accumulate in a specific non-random pattern. So if you have a truly randomized screening library, and with a lot of plates, you wouldn't expect all of the hits to basically accumulate in like one corner of the plate. You would expect them to be distributed evenly. If this is not the case, you might want to think about a different normalization strategy. Let's go back to the hit list. What you can now do here is you can select the experiment and readouts you actually want to analyze, and we're interested in analyzing all three of them together. So we select them. Then we already identified that the B-score or the robust set score are quite good normalization methods for this data set. And then we have the hit detection method. This influences the sensitivity and specificity of the hit detection. So here you can go, for example, fixed cutoffs, which is basically just defining thresholds to accept hits or not. Or you can go for something that is based on standard or median absolute deviation. So here, let's go for the more robust median absolute deviation, and a common choice here is a margin of 3. That means plus or minus 3 median absolute deviations. And then we can see here, that gives us 56 hits. We can also increase sensitivity a little bit, that gives us 94 hits. So depending on the number of hits that you want to use for a follow-up, you can play with these parameters here. So now these hits that we have here, they are mixed, so they come from different readouts. And there's one readout that we actually don't want in there, because actually what we want is to remove hits that we find in the negative drug arm, in the minus drug arm, from the other two hit lists. And for this we can use the differential screening setting here, which allows you to define a reference experiment and readout. So let's go for DAPI cell count here. And all the hits that are now found in this readout are removed from the other two. This is quite neat because it gives us exactly the hits that we're interested in. And now let's have a look at the hits plot and see what's left over. We can see we have 5 promoters here, and you can see in the hit list, actually, if you sort it, these are all from the Caspar screen. So these are the only hits that the only samples that really increase caspase activity and have apparently something to do with apoptosis. And then we have a large number of screens that stem from the cell titer fluoro screen, so they affect cell viability. And you can then also see immediately, like here we have two microRNAs that have a really, really strong effect. microRNA 634 and microRNA 491.5p. So these two are definitely worth checking out. But the question now is, well, what do all these microRNAs do to the cells? And this is something you can only answer if you look at the genes that they are targeting. So in the next step, you move over to the microRNA tab here. And what you can do here is you can look for microRNA targets by querying one of several databases that we have included. We even have some that work through web services that come from the Diana lab, and these are basically the most advanced techniques that we have here. MicroT is one of the first algorithms that actually looks at the at sequence similarity in sequence complementarity in the coding sequence, and we have Diana Tarbase, which is the largest database of experimentally validated microRNAs. We also have RNA hybrid, where we build our own database based on free binding energy. And this is particularly interesting because it allows you not only to look at microRNA targets, which actually are quite a lot. So in this case, we can find almost 100,000 targets for the 100 microRNAs that we have. So this is definitely too much. What we can do is we can play with the p-value threshold to reduce this number to something more reasonable. However, what we also have included here is the microRNA high confidence targets feature. So the idea here is, well, each gene is targeted by hundreds of microRNAs. So that's not really helpful. However, if we find genes that you would not expect to be targeted by a lot of microRNAs, then this might be meaning something. So therefore we included a hybrid geometric test feature. And you can see if we start this test, we don't get anything out of it because we are too conservative. But if we adjust the p-value threshold, we see there are actually microRNAs or genes that are targeted by several microRNAs, and we can also filter for this with quite a good adjusted p-value. That means, well, this gene being targeted by 2 microRNAs is not something that you would expect by chance. And with these results, you might already have some leads for follow-up analysis. And what you can also do here is you can look at the MirCancerDB, which contains links of microRNAs and PubMed publications that explain how microRNAs are involved in cancer. This already gives you a lot of information about the hits that you find here. And then we have one more feature, which is the microRNA family coverage. And here the idea is, if you have a microRNA family, they all share the same seed sequence and thereby also target more or less the same genes. Now if you have from a large microRNA family a microRNA that that has a very strong phenotype in your screen, but all the other members of the family don't do this, then this might look a little bit suspicious. It could then very well be an off-target effect, which is a common problem in high-throughput screening. However, if you have several members of a family that all have been identified as hits, then this might be, well, giving you more confidence in your results. So therefore we have included this feature where you can study how the hits basically correlate to the different families. But now that we have identified a number of microRNA target genes, we can move over to some more systems biology analysis. So here on the Genes tab, what you can do is classical gene set analysis. So using any of the defined gene sets here, you can on any of the 2 target gene lists, perform hypergeometric testing. However, this does not always give you results, because especially for microRNA screens where you target many different genes, it might not always be the case that you find a significant enrichment for the different gene sets. Also, we can't do gene set enrichment analysis, which is possible for RNA interference screens, for example, because, well, in RNA interference screens, this is possible because for each of the genes you have a score, and here we don't have that, so we can't do it. And as you can see, nothing really came up here. So now that we don't really see anything interesting here, we want to move over to de novo network enrichment. So here we have integrated HiTSeekR with Key Pathway Miner, which is a de novo network enrichment tool. And again, you can choose between the microRNA target gene list or the high confidence target genes as input. And here you can also select a network, for example, I2D. And what you can then select is well, 2 parameters basically. One that tells you, well, when do I consider a gene in the network as active? If it is hit by 1 microRNA or 2 or more, or all 40, which is unlikely, of course. So now we go for 1. So each gene that is targeted by a microRNA is considered a hit. Then we have the number of exception genes allowed. This basically gives Key Pathway Miner some flexibility to add nodes that are not targeted by any gene, with the goal of connecting smaller subnetworks into larger ones. So for now, let's just run with the default settings and see what Key Pathway Miner does. So you can see up here, we see the progress here in a percentage, and there's also a link provided to us. If we click on it, we are actually redirected here to the Key Pathway Miner web page, where you can see our run is currently active and running. And we can go back and see that the progress is actually identical here, and then we have to wait a little bit. Meanwhile, I can explain what border exception nodes are. Those you typically want to remove. So that's one of the problems you have when you allow for exception genes, that Key Pathway Miner basically can attach this exception gene to any of the genes that are already in the subnetwork, thereby making it bigger. Then you get a large number of solutions that are inherently identical, because the only difference is a different exception gene each time. The only exceptions that we actually want are the ones that are actually really connecting different subnetworks, and therefore we want to remove these so-called border exception nodes. Now, what you can see here is we got a result from Key Pathway Miner, and you see the gray genes here, they have been found to be targets in the network, together with some exceptions. And it's, yeah, a little bit of a hairball right now, but we can see that actually a lot of the suppressors are somehow connected in the network, also to promoters. So what we want to do now is make this more restrictive. So we are telling Key Pathway Miner, okay, a gene is a hit only if it's targeted by 2 microRNAs. And then we just run again. Meanwhile, we look at another feature here, which is called Diana-Mir path, and that's also connected through a web service. It's a website that also accepts a list of microRNAs and tells you something about pathways that are enriched for target genes. Instead of relying on the target genes that we have found, this online tool uses the microT CDS score, and you can also define a cutoff for that. Now if we start this analysis, it's not running in the background, so you have to wait a little bit and be patient. But yeah, if the number of microRNAs is not too big, usually it doesn't take too long. So maybe I should say something about the gene selection methods here. So there are different ways you can use Diana-Mir path. You can look for intersections or unions of either genes or pathways, and in this way influence how the pathways are considered a significant hit or not. And what Diana-Mir path actually reports are KEGG pathways only. And yeah, thereby it also relies on the functional annotation. As you can see here, the most significant hit is fatty acid biosynthesis, and the other ones we have here, they are related to signaling pathways like Wnt and PI3K ACT signaling and so on. Yeah, but as I said, this is basically restricted to what has been defined in KEGG, whereas the de novo network enrichment, that is, as you can see, meanwhile completed, helps you to generate new hypotheses that have not been captured in current functional annotation. And here we see, okay, now we have less of a hairball, we can actually see something. We can see, okay, this is definitely the exception gene, and the other ones surrounding it are targeted. So this might be an important subnetwork in vorinostat resistance or sensitivity. Then we can use this slider here to go through the top 20 solutions that Key Pathway Miner has found. Let's just do that. You see different results. But what we have achieved through selecting these parameters now are that the solutions are rather small. However, one feature that we have is that you can check here this checkbox, and it will HiTSeekR will actually take all the 20 solutions and build a union graph out of that and see if there are actually overlaps between the solutions. And what you can then see here is in the color green, reoccurring genes. That means genes that are part of several solutions. And the brighter the green is, the more often the solution is found. And if we now look at this, it seems to point at SF3A1, and probably means that this gene is somehow important in this. And what I now challenge you to do is to actually go to the HiTSeekR website at hitseekr.combio.sdu.dk and to select the vorinostat RNA screen, because there you should find that SF3A1, if you knock it out with an siRNA, actually causes a phenotype 2. And in this way you can use systems biology even for microRNA screens to learn how you can design follow-up experiments and how to interpret the biological meaning of the microRNAs that you have found. With that, I would like to thank you for your attention and really encourage you to play around with HiTSeekR yourself. Don't forget to enable the help function and go through the tutorial. That should give you a head start, and basically many things are also self-explanatory. Should there be any bugs or problems, please contact us per email. Thank you.